Enormous progress has been made in genomic data collection. Yet precision medicine is still stuck with the analysis process taking months and missing out on breakthrough discoveries. Until now. Alpha Vision for Medical by Aqumin offers research scientists a solution that dramatically speeds up the analysis process from months to minutes, with greater potential for discovery. Next-generation sequencing generates millions of data points, such as gene expression and sequence variations. Unlike traditional analysis, AVM instantly blends together the different types of data based on the gene. The data is then automatically aggregated by type and defined groups. This is mapped to a unique 3D visual representation, enabling side-by-side analysis of related data by the researcher. When applied to the entire set of genes, biologically meaningful patterns are revealed.
While we were speaking with VCs, everyone wondered how it was possible that we could move so quickly to figuring out which patients responded to which therapy. And the reason is because we had access to AVM.
Deka BioSciences is an early-stage biotech company developing novel therapies to drive cures for cancer and control inflammatory diseases. Only 3 years ago, Deka set out to build the next-generation cytokine platform. It created diakines, therapeutic proteins designed to deliver combinations of cytokines to diseased tissue. Each patient responds differently to each cytokine. To answer the question of what causes the different responses, Deka needed to analyze millions of data points, from experiments such as expression analysis and whole genome sequencing.
With such an enormous data set, trying to find a genetic signature is like trying to find a needle in a haystack in a field of haystacks.
The standard way to analyze these data includes heat maps and PCA plots. These reports are hard to interpret, lead to long delays, and each type of data is analyzed independently.
As a scientist, you want to be able to get back into the lab and conduct experiments as quickly as possible. And what that means is that you have to understand your data quickly and whether or not that experiment lets you know if you're right or wrong. Because the more quickly you can understand the series of correct assumptions, the faster you can actually develop a therapy to help patients.
AVM enabled Deka to formulate and check hypotheses in real time. It allowed them to explore what-if scenarios without being cost-prohibitive.
Using AVM permitted us to quickly identify the high, medium, and low responders to each of our diakines. It's AVM's ability to permit the scientist to visualize millions of data points simultaneously that permitted us to find the genetic signature associated with high, medium, and low responders to each of our therapies. And we were able to do this within minutes.
AVM gives scientists the ability to blend all biological and clinical data. To discover genetic signatures, Deka used AVM with gene expression and variations.
The initial data set was generated using IL-10 stimulated CD8 T cells, and the responses to IL-10 were predicated on the high, medium, and low induction of interferon gamma. And you can see the high response signature in the bottom left of the landscape and the low response signature in the top right of the landscape. What came out of that data set from a pathways analysis was interleukin-10 and interferon gamma receptor pathway stimulation was differentially enriched in the high responders versus the low responders. And so it was this data that actually gave me comfort that AVM is truly a profound tool for the ability to take massive amounts of data and understand them extremely quickly.
Researchers now have a tool to make biological sense of their data in a way statistics cannot. AVM's interactive side-by-side 3D analysis enables precision medicine to make discoveries faster and not possible with traditional approaches.